So, hello and welcome to another episode of CGEn's podcast series, The Synapsis, where we aim to bring together national and international experts to discuss the life science ecosystem, the current state of the affairs, and ideas that really form the future direction of science in Canada and across the globe. Today, I'm especially excited to welcome our guest, Mark Blaxter, on the podcast. Mark, thank you for joining us today.
It's good to be here.
So Mark is currently the head of the Tree of Life program at the Wellcome Sanger Institute in Cambridge, UK. Mark leads a new program that aims to generate and analyze reference genome sequences from many thousands of species across the Tree of Life, and also has active participation in the International Vertebrate Genome Project and the Earth Biogenome Project. Before joining the Sanger in 2019, Mark was a professor of evolutionary genomics in the University of Edinburgh. Mark's been based in Edinburgh for nearly 25 years. He's built a research portfolio that focuses on genomics of neglected organisms and the interpretation of those genomes in ecological and evolutionary contexts. I think I first met Mark in the UK when he was building Edinburgh Genomics, the University's Genomics and Bioinformatics facility, which went really from one instrument service to a major genome hub, servicing hundreds of customers and colleagues across Scotland and really contributing to the human agriculture and ecological genomics research. It was about the same time when I was working to establish a core facility at the University of York. And Mark had graciously provided a lot of guidance and help from his own experience. So thank you, Mark, for that once again, years ago. So Mark, I'm very much looking forward to our discussion today. It's a topic kind of definitely to your heart, close to my heart. So I'm excited for you being with us today. To kick things off, let's start at the very beginning. So can you talk a little bit about the role, what role does genomic sequence information play in conservation and biodiversity efforts of the future?
Yeah, thank you, Naveed. So I've been sequencing genomes since I was a little scientific child, as it were. Obviously, I didn't start when I was small, but even in my PhD, effectively what I was doing was a genome project, even though I didn't know it at the time. And I'm really aware that this sequence data that we generate is the common thread that runs through all of biology now, that while we can't really yet decipher phenotype from genotype, genotype is almost the best proxy we have for analyzing phenotype. So when you get to humans, we have millions of genomes. We have a huge understanding of the link between genotype and phenotype in the human. But for most other species, we have none at all. So I think that genomics is, if you like, a common language which can help us understand all life. And in the context of understanding biodiversity, I'm really interested in the diversity of life and how it came to be. And there's two bits there. There's the amazing inventions that evolution has fixed, if you like, the inventions that natural selection has generated, which allow organisms to survive in the most astonishing circumstances in the most interesting of ways. But also I'm interested in constraint. So the way that the genome sequences that we have are actually a definitive project of what we were like in the past. And so there are some instances of how you can't get there from here. And I think it's really interesting to think about the roles that constraint as well as adaptive evolution have played in the evolution of genome. So that's my interest. So I think that will help us understand why organisms do as they do today. In terms of conservation, I think it's getting clearer and clearer that in order to understand ecosystems, we have to understand the genomics of the species that live in those ecosystems, and whether it's a focal endangered species, which, you know, the target of all our interest, one species, or whether it's the ecosystem as a whole, understanding the genetic distinctiveness of those individuals and those communities and those demes, and then also understanding their connectedness to other ecosystems, groups of species, is really important. So I always like to say, just like the Human Genome Project, I mean, it was finished, not quite, in 2001, And now so much of human biology pivots on that foundational project. So much of what we understand about the human and how we, if you like, talk to each other about the human, it pivots on that genome. And I think genome sequences in the future will be the pivot, if you like, the foundation on which conservation efforts and discussions of biodiversity and the preservation of this amazing life around us rests.
No, absolutely, couldn't agree more, Mark. And I think the learning from all the efforts of the human genome and on the human side will hopefully be translated into kind of the other genomes as well, and hopefully will fast track some of those research projects and information. So that's very exciting as well. So thanks for that, Mark. So maybe, Mark, it will help the audience if you tell us a little bit about the Tree of Life project and your role within the project.
Yeah, so the Tree of Life project is one of, if you like, the 5 science departments at the Wellcome Sanger Institute. So the Sanger Institute is famous for sequencing about half of the human genome and also for sequencing many other genomes since. And so it has a tradition of sequencing genomes. And in fact, the Sanger Institute has been doing a Tree of Life project for all of the 25, 30 years of its existence, because it's also sequenced the nematode worm C. elegans. It's also sequenced vectors of parasites. It's also sequenced parasites, and it's sequenced many, many, many pathogens and a bunch of vertebrates. So it's a natural extension of the Sanger's previous delivery to branch out into all life. So in 2018, a group of scientists focused around Harris Lewin and colleagues at Davis in California proposed something called the Earth Biogenome Project. So this was a project to sequence all life on Earth, so all 1.8 million or so species. That itself grew out of the Vertebrate Genomes Project and the Genomes 10K project, which itself grew out of the Human Genome Project. So this is all a direct line from sequencing the human. And the idea there is just that these genomes will be part of securing the future of life on this planet. So not only the life of the species that are sequenced and the ecosystems, but also ourselves. We rely on all these organisms to keep us alive. They provide essential ecosystem services to us, even if we don't realize that they do. So the Sanger Institute was part of that initial discussion around the Earth Biogenome Project, and Mike Stratton, the director of the Institute, and his colleagues went to the Wellcome Trust and said, we want to start a new program at the Institute focused on biodiversity genomics. And so the Tree of Life program was born. I am in the completely amazing position of having my dream job. So I can't imagine a thing I would rather be doing than leading this amazing project. So what we have are teams of absolute experts. I would claim the best in the world, and they'll be embarrassed by that, but I'll defend them in terms of doing the sequencing. And then we are collaborating with biodiversity experts outside the Institute. We're not a biodiversity institute, to collect and identify the species for us, and also collaborating with experts in genome analysis, again, outside our Institute, places like the EBI and the Ensemble database, to provide analysis of the genomes. And we set ourselves the small goal in the initial years of sequencing the biota that live in this little archipelago of islands off the west coast of Europe, which in human terms is the 2 countries of Britain and Ireland. In species terms, that's about 60 or 70,000 species. But excitingly, despite the fact that it's wet and windy and possibly quite cold, it's actually quite biodiverse. And so 40% of all the families of organisms on this planet can be found on the lands, in the rivers, and in the seas around Britain and Ireland. So we think by demonstrating the feasibility of doing a biogenome project on these islands off the west coast of Europe, we'll demonstrate the feasibility of doing it for the whole planet. And so 3 years ago, we started with myself and 2 others. And we've rapidly built from that. So we now have a team of about 70 people here who are dedicated to the sequencing of all life on Earth. And I just say, catching, many things are easy to find. Plants don't run away, so they're quite easy. But many things we're finding and sequencing, we don't want to have to go and do that finding and sequencing again. And so we're trying to do the best we possibly can. So we're trying to do what we call reference quality genomes, where most of the DNA sequence we generate is assembled into oriented and ordered scaffolding into chromosomes. So it's not quite telomere to telomere. If you've read the literature about the recent human genome, the sequence telomere to telomere, but it's the closest we can get for this wide variety of species. So that's where we are. We're, if you like, a department in the Sanger Institute, and our goal is to sequence everything.
And Mark, just while you were talking, it came to mind, and you talked a little bit about finding these species. So how do initially you prioritize, which species to sequence? Is it whatever is available first, or is it some sort of some method to the madness, as they say? How do you do that?
There is definitely method to the madness. You all may recall the recent COVID pandemic, and I hope you're all well after it, and I hope you're all still, or your families and friends are still well. But that also forced a change in how we're working. It's only 6 months after we started, COVID hit. And all our grand plans for expeditions and field trips and collecting were basically stopped. So in the first year, I must say it's whatever we could get hold of. So there are lots of wonderful naturalists out there who, for their own sanity and mental health, need to collect. And so they were collecting in their back gardens, and they were collecting on the short walks they were allowed to do each day. So in the first years, we've sequenced far too many butterflies and moths, or just enough. I mean, they're lovely genomes to look at, and they give us lots and lots of information. And butterflies and moths are very important members of ecosystems. They're a tenth of all the species on the planet. So the fact that we've done 250 butterflies and moths is not a problem. But really, we have two organizing principles. One is we want to get one for every family in Britain and Ireland. So that's about 4,200 species. And we're trying to collect two from each family just in case one doesn't work. So that's 8,000 on the list. But the other thing is we have this lovely, you can call it a get-out clause or a mantra, which is if it's important to you because you work in it, you're a PhD student, and this is the subject of your PhD thesis, we'll sequence it for you. If it's iconic, so that means if it's got good public recognition, if it's iconic for an ecosystem, if it's a keystone organism, we'll sequence it. And if it's interesting, so if we think that it would be really interesting. So those three, that's why I call it get-out clause. We can sequence anything on the basis of that. The overall overarching goal is to sequence one per family in phase 1. And that's what drives most of the sequencing. But then we do a lot else using this important, interesting, iconic mantra.
Brilliant. Absolutely brilliant and fascinating. So one of the things, and you touched on that a little bit, is all these local projects or national projects that are kind of feeding into and connecting to each other. Can you talk a little bit about why is it important for countries and regions to have and initiate and manage their own biodiversity projects? And then how do these national local projects kind of link into the Earth Biogenome Project, which you mentioned earlier?
Yeah. So the species we share this planet with do not care about human boundaries. So there are some species which are incredibly local, and their range may lie completely within a national boundary, and they're called endemic to that country. Actually, they're just endemic to the piece of the earth they live on. Other species are found around the world, golden eagles. Okay, so golden eagles are found in the UK, they're found in Denmark, they're found in Finland, they're found all the way to the Kamchatka Peninsula and to Japan. Okay, so that is not a British and Irish species. That is a global, a species of the global Northern Palearctic. But so what we feel is there's two reasons for having national campaigns. One is it is easy to not quite sell, but persuade and engage stakeholder funders and stakeholder collectors in the project. Because there is a national element.
Interesting.
But along with that, there is this huge feeling of contributing to something global. So we've sequenced the Golden Eagle, the data are on GenBank. So you can work on Golden Eagle genomics wherever in the world you live. But that Golden Eagle genome is not it happens to be from a Scottish specimen, but it's a global species. And I think for me, that's one really important part of our project, is that all our data are open, from what specimens we've collected to what sequence we've generated so far to the assemblies, and finally the publication is all completely open. So, and it's open for reuse. It's not just openly visible, it's open for reuse. So I think that the role of national campaigns is because they have legal right under their national legislation to collect these samples. And they also have the duty, I would say, and the right responsibility to make those data public, so they can explicitly release those data publicly and globally. And by having lots of national campaigns, you build the resource, you build the expertise, you build the human resource that's needed to then deploy those data in conserving biodiversity and promoting species survival, in looking at the effects of climate change and so on. Perfect.
Thank you so much, Mark. And talking about national and local projects, as you're very familiar, we started our Canadian Biogenome Project last year. That's as Canada's contribution to the Earth Biogenome Project, led by Steve Jones, who as well you know very, very well. So I just wanted to ask you that, in your view, what are some of the unique aspects you think that the Canadian Biogenome Project may play an important role in the global context?
So the Canadian Biogenome Project is a really important initiative. It's one of the biggest in the world, and I think that's really important to celebrate, that these projects, even though they're small in terms of the number of species, but they're really important signposts on the way we want to travel. I think another important thing that I'm really impressed by the Canadian project is the way they've engaged not just the wider scientific community, and so that's not just people involved in genomics and DNA, but also people involved in ecosystem understanding, looking at ecosystem services from agroecosystems to wild ones, but also the indigenous peoples and the local communities who share their bit of the planet with these species, and for whom many of these species have very special roles. So I think for me, one of the really strong bits of the Canadian Biogenome Project is the engagement of these stakeholder communities, whether they're industry, whether they're science, or indigenous peoples, local communities in the project. So ownership, if you like, a felt ownership, a social ownership of the data is very clear from the beginning. So it's early days for the Canadian Biogenome Project. The data they have produced so far is of top quality, and the team led by Steve is dedicated to producing the highest quality genomes. In terms of biomes, the northern biome, which is obviously a focus of the so the Nearctic biome is absolutely fascinating, and is also one of the ones that is most impacted by climate change. And so it's one of the ones where, unfortunately, we're going to see the most change, but we're also going to start seeing the resilience of species to the change, and start thinking about how our planet will look in decades to come.
Perfect. Thank you so much, Mark. We can talk for hours, I'm sure, but just cognizant of the time, I thank you for your time and your input, and I'm very sure that especially kind of the younger generation who's interested in biodiversity and he wants to come and play a role in this area will listen to your podcast and be inspiring to them. So I really thank you for your time.
That's great. Thank you. I mean, what I want is for every biological scientist growing up now, and every ecologist and every biodiversity surveyor, to expect that the genome of the species they're interested in should be available, that they are a genome-ready generation. And just as I assume that PCR is possible, but actually PCR was invented during my scientific lifetime, if you like, but PCR just happens now. Barcoding just happens now. And I want it to be that this project we're all involved in, Steve Jones and the Canadian project, ourselves in Sanger and across the world in EBP, is going to change biology forever, change the way we do biological science forever.
Thank you very much, Mark. Really appreciate it. Thanks a lot.
Thank you.
Thank you. Bye-bye now.